Another way to get fingerprints or really organism identifications is a process called terminal RFLP. This is a technique that's kind of lain in the background for a long time. Not a lot of people use it, but it holds a lot of promise. And the real issue is how cheap and easy is sequencing going to get? Because the sequencing, high throughput sequencing gets easier to do individual experiments, then there'll be no need for this technology. But right now, it's a very powerful technique. The way this works is that you do a PCR reaction, and one of your primers has a fluorescent label on it. And so you get a band of a particular size. If you've done this correctly, those primers are a little bit variable, and so you may have some variation in size from sequence to sequence. But you amplify that by dividing this DNA up into separate tubes and then digesting it with restriction enzymes. And these are restriction enzymes. Typically, they're 4-base cutters, so things that have 4-base recognition sequences. And so they will cut frequently in something like the ribosomal RNA. These include things like K3, MSP1, SAL3A1, etc. And so there are going to be a variety of restriction sites, and these will be different for every sequence. And so some of that difference will be because you've got variable sequences, some of which will have a restriction site and some won't. Some of them will be as the sequence length in the variable regions change. So you'll get different patterns of bands for every sequence. And typically, people will cut with 3 different enzymes. Now, for this experiment to be useful, it's absolutely critical that you are intelligent and thoughtful about how you choose the primers and how you choose the restriction enzymes. Because the hope is that you get, for every organism in your sample, you get a different banding pattern. And so, for example, in this case, the Hay3 gives you a single very small fragment, MSP1 gives you a 571, and SAL3A1 gives you an 823. And that pattern of bands is unique to that sequence. Now, for a pure culture, then, all you would have to do is run these out on a gel, measure these bands. It's really easy in a sequencing gel to measure the size of one of these bands to the base pair. I mean, that's how sequencing gels work, right? And so you know exactly how big these are, and then you go back to a database and you ask the question, what sequence would generate this pattern? And if you're lucky, there's a single hit and you know what it is. The cool thing is, this even works in mixed populations. The trick is you get complicated banding patterns, but if there's one thing computers are good at, it's deconvoluting complex patterns. And so it's pretty straightforward to ask a computer what collection of sequences from your database would recreate that pattern or pieces of that pattern. And so this is from the paper we're going to talk about in a minute. There's 2 restriction digests. You've got a series of peaks, and you can identify the organism based on pairs of peaks that exist in those 2 different patterns. And so this is from dental plaque, and you have Porphyromonas gingivalis, Protella intermedia, Fusobacterium, some streptococci, Peptostreptococcus, all the standard issue sorts of organisms you would expect in dental plaque. Yeah.
So you take a population of sequences from some sample, and then the computer will tell you they recognize that pattern of groups of sequences.
Right. So for example, it would scan the first pattern and say, this peak is consistent with these 3 or 4 organisms, right? And then it would look for the correlating peak in the other digest. And if it's not there, it would mark it out.
So it's not looking for individual sequence patterns within a set.
No, it has to look at both patterns together, just like this. Now, it isn't going to be able to determine all of them because there are going to be some organisms in that population that you don't have the sequence for in your database, right? What that means is that this approach works best for populations where we know a lot of the organisms already. And so things like, the human gut and teeth and that sort of thing are perfect populations for this sort of analysis. Going into dirt may not be a good way to try to do this. for two reasons. One, most of the organisms aren't going to be in the database. And two, the patterns are going to be hugely complicated, right? And the more complicated the pattern is, the harder it's going to be to definitively identify the things out.
So this would be useful for if you intentionally set out to identify some sort of organisms.
No, this only works if you already know the organisms and have their sequence in your database.